you. Thank you for inviting me here. It's a pleasure to present here our national project. And as the title of my talk, the name is MiBraScan. And so we tried to develop a microbrain scanner for cerebro-vascular diseases monitoring. So somehow is similar to the project that is the one generating this workshop. So before entering in detail of the project, so to give you some insight of what we are doing within the project, I would like to thank all the people that are involved in the project. So my colleague in Politecnico, Tobono, Rodriguez, Dassano, Turbani, and Kazu. Then also the other institution that is a part of the project, that is CNR, so the National Research Center in Italy, in particular the IREA Institute, and the involved people are Scapatici and Crocco. Then within the project is also involved the University of Naples, Federico II, with Bellizzi and Tedeschi from the Department of Advanced Biomedical Sciences. And we have also a partner from France, from Paris, the Central Superlect, and the involved people are Jokamovitz and Duchenne. Okay, so just a brief outline. I will start with the motivation. I will be not long on that because the motivation are already well explained by Panos. Then I will try to give you an overview of the imaging system that we designed and now we are realizing, with some example of image reconstruction obtained first by simulation. Then I will move to the hardware prototype that we realize, and then a bit more details of how processing we are doing on the measured data, and then example of image reconstruction that we obtain from measurement, image reconstruction. And then I will spend very few words just at the end about another project that I'm coordinated that is involving also KCL, that is an European network that is focused on electromagnetic imaging or microimaging for generation for the realization of medical devices. Okay, so the work motivation has already well explained. So why brainstalk is an important disease, we know I discovered a lot of interesting things today, so I'm more convinced than before that it's important to work in that direction. It's also clear that possible imaging methodologies are a great support for clinicians. This was said this morning, and is already highlighted that the two diagnostic tools that are used mainly right now are MRI and CT, and we had a lot of details on them. The motivation behind the idea to develop a novel technique that could be complementary to the ones already available, the main point that we highlight are the following. So if possible, we would like to do an imaging with less time if compared with MRI and CT. These techniques are not easily portable. I saw an example in an ambulance, was shown this morning, but it's evident that it's not so easy to be done. They are costly, in particular, that was underlined for MRI. If it's interesting, for sure these two techniques are not available at bedside. So if there's not a machine that you have just beside your bed, and this depends, but anyhow I have to highlight that CT is using ionizing radiation. So these motivations are involving us to think about a complementary technique, so complementary to the one already available, and our main aim is to do a monitoring of the patient after the stroke onset. So what happens in the brain after the treatment that was used according to the first analysis. And so this is the kind of device that we are trying to develop within this project. The device is using microimaging technology that was all explained by Panos this morning. So essentially is related to the fact that the brain tissue, they show different dielectric properties at microfrequencies, and in particular has been shown in several publications that there is a significant electric contrast at microfrequency between the brain tissue and the stroke area if it's hemorrhagic but also ischemic. And so this is just, as I already said, the name of the project. This is a national project that was funded by our Italian Ministry for Education, Research and University. Okay, this is just a very high-level overview of the system. So the antennas of the sensor, we can call, are placed conformal to the head, like a helmet in the upper part of the head. All the antennas are connected to an antenna switch, also called a switch matrix, that is controlled externally by a PC and is connecting each time to just two antennas, just an antenna pair, to this other block that is commonly called for engineering a vector network analyzer. At more higher level, this is a device, an active device that is sending power to the antenna and is able also to receive the power received by the receiving antenna. So all the received signals from this device are given in input to an algorithm that is able to process the data, and then it comes out with an image. Okay, a bit more details about the processing. The processing is done by, in our project, at least we are using a specific algorithm that is the so-called truncated singular value decomposition, and later on I spend a few more words on that, which are the input of our algorithm. We have two kinds of inputs. First, the differential S parameters, so essentially the data that are measured by this block here, I call as differential because the idea is to do a monitoring. So we have a measurement at different time instant, and each time we differentiate between two time instant. And instead the operator is obtained from full wave simulation, and is the simulation of the system in a reference scenario, so like with an average head in the middle. This is the other information needed by the algorithm. Elaborating the data, we're able to form an image, and this is just an example of the image. We will see many more after. Okay, the first point in order to design the device is to understand which is the best frequency range, and the microwave frequency range is very wide, so which is the subband that we should use, and also in where we place the antenna that is around the head. The antenna could be in air, or we can put all around the antenna a proper medium, the electric medium, that could improve the performance of the antenna. Essentially, improve the performance means to maximize the power delivered inside the head. That is obvious, that is what we want. Okay, I report here this very nice diagram that is taken by these references, where is done a relatively simple, but we found powerful analysis. So what is done is to consider all the different tissue that are within the brain, so starting from the skin, fat, bone, and so on, and just to do a 2D problem, so like a multi-layer structure. And then here there is the antenna, so the incident field within the matching medium. What is reported here is the so-called transmission coefficient, that is a measure of how much power is able to enter inside the brain, so it's not reflected by all these different tissues. And this plot is showing here the frequency, okay, what is changing with the frequency, and what is changing with the medium, the external medium, the so-called matching medium, where the antenna is placed. The region where we want to work, so where this coefficient is maximized, are the red one. And so we see there is a region here that is the one that we have chosen. In particular, we will work around 1 GHz with a medium, matching medium of around 20. 20 is the relative permittivity, relative electric constant, the electric constant. Here in the middle, instead, there is a region with a minimum, so is not suggested to work in this frequency, even if you change the matching medium, because the power is not maximized. And then you see again red here, so we can work also at higher frequencies. But the point is that we need to penetrate the brain, so they arrive from one side to the other side of the brain. And if you increase the frequency, here there is also an increase of the conductivity of the tissue. They translated, in our point of view, there are more losses within the brain tissue, so the signal that we receive is too weak in order to be then elaborated. So this is our choice, as already said. And then a few more words about the imaging algorithm. We are working with the differential imaging. So we measure the scenario at one time instant, and then the same scenario as another time instant, and we make the difference. Thanks to the fact that the difference should be located in one portion of the head, we can linearize the model that is behind using this Born approximation. Why this is nice? This is nice because the measured data, that is this delta S, is just related to the unknown, that is the variation of the contrast, through a linear operator. And working with a linear operator is much easier than working with an operator that is not linear. And this delta C, called here variation of the contrast, is what is different between the two scenes. So the change in the region where there is the stroke area between different time instants. So this is what we are trying to find. Okay, so how is work the algorithm? So this is just reported the equation. So what we do, this is more technical, so I will just to mention it. So we apply the SVD to the linear operator that is known, and so we have the singular vectors and singular value, and so we can construct, so we can obtain the unknown, that is the variation of the contrast, in a simple way. So it's just a linear combination of the right singular vector of the SVD of the operator. These are the coefficient. The coefficient is nothing else than the measured data, differential measured data, projected on the left singular vectors, weighted by the singular value. So it's an operation that is relatively fast. So the processing time in this specific kind of algorithm is not an issue. It's faster than the measuring time. Okay, so then which is the configuration that at the end we have chosen? I already said the frequency, the matching medium, and then we place 24 antennas around the head. Details of why 24, that is compromised between different parameters, are reported in this paper, pretty recent paper, where we give some guidelines of choosing how many antennas you need in order to do a good imaging. As already said, each antenna is a transmitter and a receiver, and then we need also to do some full wave simulation, and we usually use our own software. Okay, these are, so these spheres here are representing the stroke area with different sizes. This is just another view. Okay, this is a bit more in details the imaging system. So each block is an antenna. So the block is the matching medium, and inside there is the antenna that is a relative very low cost antenna. It's just a printed monopole. Here there are all the details of the coupling medium, the brain tissue, the stroke, the epsilon r, the conductivity at 1 gigahertz. Okay, they are taken from available literature on that. And so these are just some enlargement to show what happens. So each block is an antenna. As I said, the block is a dielectric. The antenna is inside. You see it now here. And then you have the brain and the possible blood sphere. Okay, first analysis, this is a very engineering analysis, but is important, is to be sure that the antenna is working well. So what is reported here is the input reflection coefficient of the antenna. Essentially, if this quantity is very low, means that the power is not reflected by just the antenna, but is tried to enter inside the brain. And so around the 1 gigahertz, that is here, the selected frequency, we have a good matching. So this is an important test, obviously, to be done. And then here there are some examples of the data that are then that can be measured. This specific one are simulated, but these are the data, the delta S, that is in input to the algorithm. This is for two different frequencies, so 1 gigahertz, 1.5, and here we are considering the two rows, two different cases. So this is the differential data between a head without the stroke area, and then with the stroke here, that is a sphere with a diameter of 10 millimeter. And this is instead with a bit larger one, so the diameter of the blood sphere is around 20. This is okay. This is a little detail. So these are the distribution of the singular values, because this is part of the algorithm. And then using this data in input, these are some example of images that we are able to reconstruct. So this is the smaller target at 1 gigahertz. The one show the red lines are showing exactly where is the ball and also the size. The yellow part is what is reconstructed by the algorithm. This is changing the frequency as expected. The higher the frequency, better is the resolution, but we have to keep in mind the limitation that I initially said about the region where we can't work because there is an intrinsic reflection due to the different tissue of the brain. This is combining. We can also use a multi-frequency data, just to, it's not so multi-frequency, but this is feasible. And this is, the example you see here is without noise. Then we try to add noise. The noise is added to the measured S parameter, so to the input data, given in input to the algorithm. First case is without noise, so what we already seen, this is for the smaller target, and then increasing the noise. Obviously, if the noise is too high, we are losing the data. With the noise signal to noise ratio of 70 dB, we are still able to see the right position and size of what we are searching. Then we tried, this is another examples where instead we are measuring a 2-time instant where time 0 is the head with the presence of a stroke area, hemorrhagic area of 20 millimeter size, and the other time instant is 40 millimeter size. So we are trying to image the difference between these two. So the correct area that we try to image is the one highlighted here between these two circles, and what you see with the different colors inside is what the algorithm is able to image, still working at a relative low frequency that is the 1 gigahertz. This is without noise, and this is adding 80 dB noise that is obviously is less precise than this, but is still feasible to recognize the extension. Okay, then we tried with a more complex problem. Before it was just an homogeneous head. Here we consider a much more realistic, so an heterogeneous head model where we have all the different tissues. Different colors means different tissue in the model. Here we have the different tissue and the corresponding dielectric characteristics, everything at 1 gigahertz. And then again we place stroke, hemorrhagic stroke of 2 sizes that corresponds to 2 different time instants in the realistic scenario in 2 different positions. We apply the same procedure, the same algorithm, and this is what we get. Of these images are we are generating, as before I didn't say that, we are generating a 3D reconstruction, and these are just the cut in the 3 main planes. And again, in black is highlighted what we are searching, and the yellow part is where the algorithm is trying to do the reconstruction. And this is for 2 different, all these measurements, these simulations, sorry, are with the noise, and this is the level of the noise. This is in one case, and this is in another position. So the same simulation, just moving in another position, the presence, the possible presence of the hemorrhagic stroke area. Then we move to the hardware. So this is what I showed until now, our simulation. Then we move to the hardware. This is just an overview of the hardware that we have. Everything is controlled by a PC. This stuff here is the matrix that is able to connect different pairs of antenna with the electronic. This is the network analyzer, so the device that is sending the power, receiving the power. This is a lab version that is not small, are available relatively small now on the market, so this is something that is not a challenging now. This is the place where there are the antennas, so this is the tank with the liquid, and here we have all around the antennas, and in the middle there is the phantom that I will explain more later on, that will represent the brain and the presence of a stroke area somewhere. Okay, few comments, this again more engineering, on the switching matrix that we realized using electromechanical coaxial switches. We use that because they are the switches with the highest isolation available on the market. Probably is more than what we need, but we must be sure that is the possible problems are not related to this part, so we use the best that we have available. They are connected with the semi-rigid coaxial cable, again to avoid communication between the different interference between the different channels. And at the end, this matrix is a 2-port, is where the network analyzer is connected to, up to, and then these are the input, and then the other side we have the 24 ports. So these black cables here are connected to the antennas. To give an idea of the measurement time, to generate a 24 by 24 scattering matrix, so the whole measurement, at one frequency with the not so optimized algorithm to control these switches, we need around 4 minutes. So for 3-4 minutes. And this is another detail. Then another important point, we will work with the weak signal, because we work with the difference between measurements, so we must be sure that we have not significant losses along the system. And so here we measure again the transmission coefficient of the scattering matrix. So this is a measure of how much power I'm losing from the signal that is entering and the signal that is coming out, including also the flexible coaxial cable. And at 1 gigahertz we lose around 1 dB. And so this is, seems to be good. Okay, more details about the coupling liquid, the matching liquid, in order to maximize the matching between the antenna and the target. And here are reported the electric characteristic, the relative permittivity and the conductivity. As 1 gigahertz, that is here, you can see that the relative permittivity is between 20 and 25, as we said at the beginning with the initial analysis. And the conductivity, obviously, conductivity should be as low as we can. This is what we obtain with this matching liquid. It is a very simple one. It's just a mixture of water and Triton X. That is, again, something easy available on the market. This is the antenna of the behavior of the realized antenna. So this is the measured of the measure of the S1 now, the input reflection coefficient. Again, you can see that at round 1, we have a good matching. So the value is low. And this is, by the way, the realized antenna. Then a few more words about the phantom. The system that we already realized is, for now, a 2D system. So we use a 2D phantom that is modeling like a cylinder. Ideally, should be an infinite cylinder. So is a cylinder with a dominant length, 8 here. Inside, you see there are some holes. In these holes we can place these other cylinders. These are representing the brain. So in other words, these cylinders are filled with a mixture of liquids, again Triton and water with different percentages, that are representing in the frequency range that we are considering, in particular 1 gigahertz, the dielectric characteristics that are known for the blood. And then externally, in this other cavity, is filled with an average value that is representing the brain. Again, the value is taken from literature. And outside, we have the matching liquid. And then just a few details about the data processing or the measurement, something that obviously is not needed in simulation, but we need in measurements. First of all, we apply a time gating. Why we are doing that? Because we have a tank, and we have a signal that is going from one antenna, the transmitting one, to the receiver one, but the signal is going everywhere, and so we start having reflection within the tank, and we don't want to have them. If you look in time domain, this is time domain, the dominant one is the one that you want to select, and these are the multiple reflection. So this can be easily removed. So we work in the time domain, we apply a window, that is this applied here, to select just the signal that we want to use. This is the phase is unchanged, and what we get, and we will see now, comparing the initial measurement, the raw data, the blue one, the red one is following the same, but is much smooth. And in general, any algorithm, imaging algorithm, is much more happy if the signal received is smooth. Then another thing that we are doing is the so-called embedding. So ideally, what we want to do, we want to measure what is happening at the 2 antenna ports. But in the middle, we have the switching matrix. So we can do that even if we cannot directly connect our network at the antenna ports. And so here is just a schematic of the whole system. So in the middle, here is the tank. Okay, here we have one antenna, the other antenna, and in the middle we have our device and the phantom and the liquid. Here is one branch, one path of the switching matrix and the other path of the switching matrix. What we are doing, we are measuring between this point and this point, B and B primus, but we want to know what is happening between A and A primus. This can be easily done, so we characterize the whole system. This is exactly what we measure with the network analyzer. Then we characterize the 2 paths, and then we apply some properties that relates the scattering matrix of what we are measuring, so that at the end we are characterizing just the 2 antennas within the tank. What is happening to the signal? So means that now we are measuring when the signal is starting from antenna arriving to the other one. So now time 0 is here, so there is this time that is the time needed by the signal to arrive to the antenna, and now here instead we are at the receiver antenna. So applying the bending technique, we particularly in the time domain, we shift the measured data. So now time 0 is here, and if we look to the frequency domain here, we have more or less the same signal, but is shifted up. Why is shifted up? Because we have no more included the losses due to the switching matrix. But you can see that we have still a signal with a lot of variation, because this is due by the multiple reflections that is not corrected by the de-embedding. Then we correct the phase, that many times is very important for an imaging algorithm. So this is the original measured phase of the signal. This instead is the one in green, is the phase that we should have if we connect our network analyzer directly at the antenna ports. And the black one is the one that you obtain after the de-embedding. You see that there are some shift here, but these are multiple of 2 pi, so the two lines are the same. But as I said, there is still multiple reflection, so as intuitive, we apply both approach together. Applying time gating and embedding, this is what you obtain. So we apply, this is the signal after the embedding, we window the signal, and so the line that you see here in pink is the real signal that is elaborated by the algorithm, that is after the embedding and time gating. And here is the corresponding phase that now is more clean, so you see there are just jump that are multiple of 2 pi, so the two phases between what we are giving in input to our algorithm and just connecting the system and the two antennas are coherent. Then example of image reconstructions with this system. So this is what is representing the brain. The cylinder inside is instead the one filled with blood, and the differential measurement is with the target in and without the target. And here you see a sequence of images that now I try to explain. The first row that you see here is, the first column, sorry, is raw data, so without any data processing. The second is applying time gating, the third one applied embedding, and the last one applying both of them. And then the row corresponds to different frequency and a little change in the operator. So we can just look to the first one, okay, it is a bit lower frequency, where the operator, that is the other data that is needed to the algorithm, is just an homogeneous coupling medium, so it's not related to the specific phantom, so the specific head that we have inside, and so it's very easy to be simulated and to be inserted. So this is the initial reconstruction. So you see the red circle is representing the exact position and size of the target. This is just a 2D system, so this is just a slice. And the yellow are the peak, the region recognized by the algorithm. With the raw data, we have some noise around, some side lobe, I could say. Applying the time gating, so avoiding the multiple reflection, the reconstruction is much more clean. Applying the embedding only, we have not a big change. And we think about that, and probably the reason is that in the way, because the switching matrix was constructed by us. So we construct the switching matrix in a way that each path, independently of which one is selected, it has the same length. And so essentially, except for error in constructing the system, that embedding is not so necessary. So this is also shown by the experiment. And then here applying both, so more or less we have the same as the time gating. The second row, we have a bit higher frequency, and we have a bit better detection and size of the object. And the last one, we have a bit higher frequency, and we improve the additional information given to the algorithm. So now the so-called operator is not just a matching liquid everywhere, but inside we consider a phantom, without the target obviously, filled with the material that we have in reality. And you see, in particular, if you look to this reconstruction here, that now there is a very good detection and also a good size with the measurement data that we use with the system. Okay, so to conclude this part, I try to describe the design and the realization of the imaging system. I show you some area reconstruction in the 3D scenario using simulation, and reconstruction in a 2D scenario with our 2D system prototype. The next step is to move to do 3D reconstruction with the measurements, and so to build like a helmet. Here is the stage, we are almost at the end of realizing the hardware. All the antenna will be placed around the head, as we already did in simulation, and so to go on with the measurement, image reconstruction using head phantom that could be also not just homogeneous, but considering the different tissues. And if I have still some minutes, okay. I would like just to mention that I said at the beginning a European project that I'm coordinating, that is a pretty wide project, is a training network The training network is putting together all the most active engineering groups in electromagnetic technology, so microwave imaging applied to medical problems. And the main aim of this network is to form researchers, so we form PhD students, at the end they will get the PhD, that in our view should be able to accelerate the translation of this technology from the research bench to the patient's bedside. That we already know also with the previous experience with breast cancer detection or monitoring, that was a bottleneck in the evolution of the technology. This is the consortium coordinated by Politecnico di Torino. All the beneficiary, as I already said, also King John's London is involved in. We are all around Europe. And then there is also the partner organization still connected to the network that has pretty wide number, as you can see. They are academic institutions as well as industries, and also a highlight here for hospitals that are also connected to the network, because even if the PhD students that will be formed by the network are engineers, it's very important to have a connection with the clinicians, as also already evident by today's workshop. And so there is the University Hospital in Bern, Switzerland, Hadassah Hebrew University Medical Center in Israel, Lariboisier University Hospital in Paris, and Hospital Daluz in Portugal. So this will be our clinical side that should help us in understanding what is needed, which are the real requirements from a medical point of view. Then the network has several general objectives. The first one already mentioned is to translate the research from just the research activity to the clinical, and this is in several parts. We will work on the technology to standardize the procedure and to develop prototypes of devices. And then there are other general objectives that are mainly related to the formation, to the training that our student will obtain during the duration of the project. So we hope to give as first, at least in Europe, a complete scientific and training program in EM medical imaging. And we hope to expose our ESR to academic, but not only academic sector, so also industry are involved in, and to give them a proper, absolutely excellent training, scientific training. And then entering a bit more in details of the first objective, that is the more scientific one. This is declined in 5 sub-objectives, and these are essentially the activities that we will done within the network. The first one is related to the idea to have some standardized phantoms, so that represent the different parts of the body, and then can be used from the same phantom in different systems in order to easily compare the different systems, and they should be significantly complex in order to really try the performance, test the performance of the system. And together with that, we need also to have correct information about the end property of the human tissue. So this is also part of the project, so to do the measurements on human tissue of the dielectric properties. Then there is another objective that is developed more the hardware, so new components that will be part of the prototypes. Another objective related to the software, so to have a proper modeling of the system, and also to develop the advanced image formation algorithms that are essential, because are the algorithm that is taking the measured data and generate the image, that could be hardware accelerated in order to reduce the time. And then we will create prototype devices for medical diagnosis, clinical follow-up, and image-guided treatment. And so this is just, as all project we have working package, it's just the same objective organized in working package. What I would like to highlight is simply that all the activity are strictly related. So if you build a prototype, you need the hardware, so you are in contact with the people making the hardware. You need the software, so you are in contact with the people making the software. You need the phantom, and so the phantom is provided by the group developing the phantom. So there is a strong interaction within the network. This is a more detailed description of the task, and that are declined according to what is said at the beginning. So activity on the phantom, on the hardware, on the software. And here in particular there are the device, the prototypes that we are developing. In Torino, for example, we still work on cerebrovascular disease imaging. So it's somehow the extension of the national project that we are currently now. in Portugal to a system that is able to do auxiliary lymph node diagnosis. In Turkey, they are working on a system for chemotherapy monitoring. In Germany, for hyperthermia treatment monitoring. And again in Italy, in CNR, for a system for image-guided microwave ablation for the monitoring of the ablated region. And so to conclude, if these are just the contacts for the European network, and we are happy to receive comments or to be contacted. So thank you.
Thank you very much. Maybe one or two questions, if there are any.
Yes.
Are you gonna use any animal tissues to test your system?
Yeah, this the idea within the project, the European project, is the following. There is a group, in particular the one in Galway, that is doing the measurement, and that are in order to understand the correct, as possible as correct, dielectric properties, and they will work also on animals. Then these are used in order to prepare phantom, that are not animals, that are just prepared based on that measurement. So what we will put inside the system are not animals, but are phantom, where characteristics are based on the measurement that are done in Galway.
Okay, and one more thing. I don't know if I missed it. I think you said you are transmitting with an antenna, one antenna, and receiving with one antenna.
Yeah, and then we switch between all the
Yeah,
antennas.
yeah. So have you, I mean, I was just wondering if you try transmitting with one antenna, but like receiving with multiple antennas at with close proximity to see if the image quality is increasing.
Yeah, essentially we are doing that. Well, only switching. So we have one antenna transmitting, and then the receiver antenna is switched. So we don't do at the same time, but in the same situation, hopefully. And so we have a multi-view of the system. So we do what you are saying, but not at the same time. So we switch, and so we do the measurement with a multi-view. So one transmitter and all the receiver around. And then we switch another transmitter, all the receivers around.
Yeah, I understand that. I was just wondering, because if you send one, because when you send and you get the reflection,
yes.
So the same reflection could probably give some better idea when you have different angle of, let's say you send this direction, you just get from that one direct, but that one, it leads to other reflections. Probably you might have some other.
But the transmitter is always the same. So you have a transmitter that is somehow fixed. Then I'm receiving here, and as you said, the signal is going not only there, it's going everywhere. Then this is no more active, but it's just a receiver, so it's not sending power. So you have another antenna here that is taking what is happening in this direction, and so on. So I think that I mean, it's different time instant. So if something is changing with time, I agree with you, that should be done at exactly at the same time. But imagine that in the measuring time that are talking about fraction of milliseconds, so just one scan with one antenna transmitting, we expect that there is not a significant change in time, and so we are able to do the multi-view measurement that is essential. Without that, we are not able to reconstruct.
Okay, thank you.
Yes.
Just a brief comment about your exchange. I think that as far as you have the individual response to one antenna, you can, by superposition, simulate simultaneous excitation. And I think that's a good model what you were suggesting.
Okay. Thank you. Any other questions or comments? Yes.
So as we know, this phantom or human tissue is very lossy. So if you rely on S21, do you think if the signal bypass, instead of transmitting to the tissue, if bypass,
Yes.
how you solve this, like how you try to concentrate?
Yeah, the point of this, I think, is the best to show what we have seen at the beginning. Okay, we have two effects that are together. Here we are showing one effect. It was at the very beginning. Okay, so we have losses in a more general view. So losses that are taken by the tissue, and losses because you are reflected away when you have this dielectric discontinuity. For our point of view, are both losses, so our signal is not arriving. So if we think about just the fact that we have the interface between different dielectrics, this is the analysis that was done. So is essential, is having, this was not expected at the beginning, but here is This is just a 2D simulation, but was replicated in the second citation with a measurement on a sphere with all the different tissue, and this is more or less the same result that is shown here with a simpler case. And so this is already selecting us some frequencies. So we work at 1 gigahertz or higher than 4. The second, your comment about the losses in the tissues, this is related to the fact that because we are not choosing 4 or more than 4 gigahertz. Because in that case, you will be very happy about resolution, higher the frequency, higher the resolution, but the losses will dominate. And so the signal that we will receive, and consider that we work also with differential signals, so the signal that is just due by the target, that could be relatively small, is so low that is in the noise of our receiver, so we don't see it. So with following this first analysis, we work at around 1 gigahertz. That seems to be reasonable in resolution, according to the fact that the antenna, by the way, is not in air, is in a matching medium with a relative high epsilon r, so this is improving a bit the resolution. And the signal is not, I mean, we are not talking about high signals, so I give you, again, here there are some idea of which signal we are working with. But this is something that can be detected by network analysis. So, for example, this case is 1 gigahertz, okay, with a target from what we are able to do, relatively small, so 10 millimeter size. This is a 3D target, so it's just a sphere. If you put a cylinder with that size, is a stronger target. And the signal is around minus 90, minus 100 dB. So it's not a strong signal, but it's something that you have a good network analyzer with a good dynamic range, you are able to detect.
Okay. Thank you very much again.
Okay.